Welcome back to IGCSE Biology Revision Series. Today, we will be diving into Chapter 4 Biological Molecules, the building blocks of life. From the food we eat to the DNA that makes us unique, biological molecules are the heart of every organism. By the end of this video, you will understand what makes up carbohydrates, fats, and proteins, and DNA, and how we can test for these essential molecules. Let's get started. Before we delve into the biological molecules, let's see a key concept. Large molecules are made from smaller molecules. We call the large molecules as macromolecules or polymers, and the smaller building blocks the monomers. For example, glucose is the monomer of carbohydrates, amino acids is the monomer of proteins, and glycerol and fatty acids are the monomers for fats. The first biological molecule we will see is the carbohydrates. Complex carbohydrates are built from simple sugars. Specifically, starch is made from many glucose molecules joined together. Plants use starch to store energy. Glycogen is also made from glucose, but it's an animal version of the starch. Your muscles and liver store glucose as glycogen. Cellulose is made from glucose too, but the bonds are different. This gives plant cell walls their strength. As we learn about the different carbohydrates, we need to also remember the three elements that carbohydrates are made of. They are carbon, hydrogen, and oxygen. Carbo means carbon, and hydrates means water, which has the elements hydrogen and oxygen. In the next slide, you will see carbohydrates forming in action from glucose molecules. The second biological molecules we will be dealing with is the proteins. They are made from building blocks called amino acids. There are about 20 different amino acids that can be arranged in millions of different combinations, like letters forming unique words. Each unique sequence creates a protein with a different function. Proteins are more complex than fats and carbohydrates. They contain four key elements: carbon, hydrogen, oxygen, and nitrogen, and sometimes sulfur. A single amino acid chain is called a peptide. Many peptides join to form polypeptides, which we call as proteins. In the next slide, you will see proteins forming in action from amino acid chains. The last biological molecule is the fats. The solid form of any fat is called fats, and the liquid form of any fat is called oils. They are made of two components, fatty acids and glycerol. Fatty acids are long chains of carbon and hydrogen. Glycerol is a small molecule that acts like a backbone. One glycerol molecule combines with three fatty acid chains to make a fat or oil molecule, and that's why fats are sometimes called triglycerides. Fats and oils, which we also call lipids, also contain the same three elements as carbohydrates: carbon, hydrogen, and oxygen. In the next slide, you will see how a typical fat molecule forms from three fatty acid chains and one glycerol molecule. Now let's explore DNA, also known as deoxyribonucleic acid, the molecule that stores all genetic information. First up, let's look at the structure. DNA consists of two strands coiled together to form a double helix. Each strand contains chemicals called bases. Bonds between pairs of bases hold the strands together, like the rungs of a ladder. Talking about the pairing of bases, there are some rules which are crucial to remember. The bases always pair in this same way. There are four bases: adenine, thymine, cytosine, and guanine. Adenine always pairs with thymine, and cytosine always pairs with guanine. Remember, A pairs with T and C pairs with G. This is called complementary base pairing. If you know the sequence of one strand, you can work out the other strand. Now, why is this important? Well, this base sequence is like a code. It's the instruction manual for making proteins. Different sequence of bases code for different proteins, and those proteins determine everything about an organism, from eye color to how tall you grow. The beauty of the double helix structure is that it can unzip down the middle, allowing the cell to copy the DNA when it divides. Each base finds its complementary partner, creating two identical DNA molecules from one. And now, let's skip the theory and move on to some practical knowledge. Food tests in detecting biological molecules. In your practical exams, you need to know how to test different biological molecules. Let's go through each test carefully. The test 1 is the iodine test for starch. This is the easiest test to remember. Add a few drops of iodine solution to your sample. If starch is present, the iodine changes from brown to blue-black. No starch, it stays brown. Simple. Test 2 is the Benedict solution test for reducing sugars. Reducing sugars include glucose, fructose, and maltose. Here's what you do. Add Benedict solution to your sample. It starts blue. Heat the mixture in a water bath. If reducing sugars are present, watch the color change from blue to green, to yellow, to orange, and finally to brick red. The more sugar present, the further along this color scale you will go. The third test is the Biuret test for proteins. The Biuret reagent contains copper sulfate and sodium hydroxide. Add it to your sample. If protein is present, the solution turns from blue to purple or lilac. And no protein, it stays blue. Fourth test is the ethanol emulsion test for fats and oils. This test is a bit different. You need to dissolve your sample in ethanol and shake well. Pour the mixture into water. If fats or oils are present, you will see a cloudy white emulsion form. The cloudier it is, the more fat is present. The last test is the DCPIP test for vitamin C. DCPIP is a blue solution. When you add a sample containing vitamin C, the vitamin C reduces the DCPIP. The blue color fades or disappears completely. The faster it decolorizes, the more vitamin C is present. This is great for comparing vitamin C content in different fruits. And that's a wrap for Chapter 4, Biological Molecules. You now understand the building blocks of life, from simple elements to complex macromolecules. You know how to test for different biological molecules in the lab, and extended students can explain the remarkable structure of DNA. If this video helped you, give it a thumbs up and subscribe for more IGCSE content. Drop any questions in the comment below. See you soon in the next video.